So I'm going to give you a very brief overview on what stem cells are. That means this is not everything you always wanted to know about stem cells, because it has to be short. But I definitely encourage you to take advantage of the stem cell enthusiasts that are in this room today to make questions and to really try to understand what stem cells are and why they are so important for society. So what is a stem cell? So biologists have defined a couple of essential properties that define stem cells. One of them is self-renewal. That means stem cells have to be able to divide, being able to generate cells that are identical to itself. They have developed a couple of different strategies, so cells can divide symmetrically or asymmetrically. But the important thing is a stem cell can maintain the pool of stem cells identical to itself. The second property is differentiation. So obviously, you can see your body. You're made of thousands of different types of cells that have different morphology, different function. So these cells somehow derive from stem cells that are able to generate these very specialized somatic cells that are different from stem cells. So these cells don't have the same properties of stem cells. So where do all stem cells come from? So there is one so-called totipotent stem cell that is the fertilized embryo. By definition, this cell is able to generate all the tissues that compose your body and the tissues that are required for an organism to have birth. So this cell can give rise to every single tissue that you have in your body. And then there are pluripotent stem cells. Now, we are going a bit down in the hierarchical tree of stem cells, so that means they have lost a bit of power. Pluripotent stem cells cannot do everything totipotent stem cells do, but they are still able to generate every single tissue in your body. Just some tissues are not being generated from pluripotent stem cells, but we will see it after. And then finally, there are multipotent stem cells. You can imagine these cells are even further down in the hierarchical trees, so they are even more restricted in their potential to generate different types of tissues, but still they have the potential to generate different types of tissues that are the specialized tissues we mentioned before. So where can you find adult or somatic stem cells in your body? In a surprisingly wide array of tissues. So you have stem cells in your eye, you have corneal stem cells, you have retinal stem cells, you have stem cells in your brain, you have stem cells in your blood. There are many cells, many different stem cells in every different tissues. These stem cells are somehow rare, and we will see how we can deal with the properties of stem cells for our studies. So why stem cells are important? What do they do in our body? Well, this is just a dramatic example of the potential of stem cells. Every second, an average person destroys something like 2 or 3 million of red blood cells. So you would be anemic by the end of the day if the depletion was just constant without a constant replenishment. So stem cells are there to replenish the pool of cells that are constantly destroying in your body. So how do they do that? You don't have to imagine that the stem cell is just constantly dividing and producing one stem cell and one red blood cell, because that would be a tremendous stress for stem cells. So we said that stem cells are rare. These somatic or adult stem cells are rare. So how do we maintain such a huge number of cells constantly dividing? Nature is smart. So the stem cell will be able to generate another cell that is called the progenitor cell that is not as powerful as the stem cell, but has still the potential to differentiate and to proliferate a lot. So the stem cells are rare. They are protected in their niche. They divide sometimes. They're kind of quiescent, we say. And then they generate these progenitors. And then the progenitors have to expand a lot. And in this way, the stem cell pool is maintained. And all your somatic differentiated cells are replenished upon demand. The second most important thing that the cells do is to repair, for example, damaged tissue. You are all familiar with wounds that you can have in your skin. It's surprising how fast these cuts heal. This is due to the fact that stem cells residing in the epidermis feel that there is a damage. So they proliferate. They migrate to the site of the wound. They produce new cells that will replenish the cells that are damaged. And then once the emergency is gone, they go back to quiescence, and they stay there waiting for the next occasion to be helpful. So we have defined now adult stem cells as multipotent. That means that they can generate multiple types of tissues. This is an example. This is the so-called hematopoietic stem cells. As you can see, it can generate many different types of cells. You can see erythrocytes or red blood cells. We said they stay around for like 100 days, and then they get destroyed, so they need to be constantly replaced. Platelets, even a shorter lifespan, still getting replaced. BET, natural killer lymphocytes, macrophages that are the cells that engulf the pathogens and the bacteria you see in the movies, and all the other cells. So all these specialized cells come from one single hematopoietic stem cells that is multipotent. So remember, toti, pluri, multi as a hierarchical tree for the power of stem cells. So how do we study stem cells? So first of all, we need to be able to isolate the cells. That means if you take a bone marrow sample or if you take your blood, you will have billions and billions of different cells that are specialized cells. So you need to be able to pick the cells that are the so-called stem cells. Biologists have developed markers that enable scientists to identify and to pull out from this soup of cells the stem cells. And then bring them to be single cells in order to be able to study them at the clonal level. That means you should be able to put one cell in a confined environment, wait for it to proliferate, and in this way you will see that the stem cell gives rise to a progeny that is identical to itself. So this is the self-renewal property we discussed before. And then the second thing, proliferation is not enough. Cells have to be able to display differentiation. So there are assays now that are available that we can perform in vitro, where we take the stem cells that we have isolated and we differentiate them into the different types of cells that they can generate. And this is a different type of stem cells. If you remember before, we discussed about the pluripotent stem cells. These are the so-called embryonic stem cells. Where did they come from? So in the first picture, you see an embryo, like an egg being fertilized. So the fertilized egg is the zygote, and the zygote immediately starts to undergo some divisions. Within a couple of divisions, the cells that you are in the screen are still totipotent. Totipotency, however, is lost very fast, and after more divisions, you get this structure that is called the blastocyst. And what you're interested in is the bunch of like reddish cells that you see at the bottom. That's the so-called inner cell mass. You can take the inner cell mass, and from there you isolate what we currently use in the lab, the so-called embryonic stem cells. These cells are pluripotent, so just to remember, they are able to generate any kind of tissue in your body.